In the last class, we discussed about the definition of bioinformatics, and also we discussed about how the bioinformatics emerged as a separate discipline in biology, and also we discussed about the preliminary concept in molecular biology, like the DNA structure, gene structure, and the codons and others. So we proceed further today in the same line. First of all, we shall be discussing about some of the remaining concept in bioinformatics and what is the role of bioinformatics in biology. How does it help us in understanding the complex, diverse biological phenomena? And as example, we shall also take on some of the important areas in biology, how bioinformatics has helped us in solving the issues related to complexities in biological system. So as we discussed earlier also, as we know that genetic code is almost universal, except in few cases where the codons differ from each other in specific species, but there are very few exceptions in that. In most of the cases, in 99.9% cases, the genetic code is nearly universal, irrespective of whether an organism is a prokaryote or an eukaryote. The genetic code remains same, except in very few cases. So this universality of the genetic code has made it possible, therefore, to transfer and translocate genes from one species to the other. That we now say as the recombinant DNA technology. So has it been not universal among different species, it would not have been possible to transfer genes across species and express them. One of the first successful transfer of genes from one species to the other was documented by transferring gene from an insect, what we call as firefly. As we know, the fireflies used to flourish during night. So from that, the gene responsible for fluorescence in firefly has been detected, and by extracting the gene sequence from firefly, when it was introduced and expressed in tobacco plant, the tobacco plant also started flourishing at night. So this is one unique example of transferability of genes across species because of the universal nature of the genetic code. Now, although so far we have discussed about the nature of genetic code and the variation caused by the changes in the nucleotide sequences that causes variation in the phenotypic characteristics in diverse species or within a species. But the genome or the gene sequence cannot explain all the variabilities or diversities in living system. Let us take a classical example of an insect. An insect has got 4 different phases of the life cycle, and the morphological and physiological characteristics of each of the morphological stages of the life cycle are different. Say, for example, these are the 2 stages of the life cycle of the same insect. One is the larva stage and the other is the adult stage. So except for the similarity in color little bit, we don't find any similarity between the 2 different stages of the life cycle of the same insect. Similarly, other 2 stages are the eggs and pupa. They are also totally different morphologically as well as physiologically. However, if we isolate the genome of the different stages of the same organism and go for sequencing, we find throughout the length of the genome exactly the same sequence. the genomic sequences in 4 different stages of the same insect will be exactly alike, 100% same, identical. But even then, physiologically or morphologically, that means phenotypically, they are quite different. So that means the overall diversity in phenotype cannot be explained by the diversity in the genotype. So the sequence diversity in the gene or genome cannot explain all the variations or diversities that we see in biological system. So beyond genome, there must be some other factors which govern this kind of diversities or variations in the living process. So there lies the role of the proteins and proteomes. As genome is the pool of genes present in a particular organism, proteome also is referring to the pool of all proteins expressed by an organism under the influence of a specific condition at a given point of time. That may be variable from time to time, that may be variable from the stage of growth, between stages of growth, or that may be variable because of the influence of several intrinsic and extrinsic factors. So this proteome variation is dependent upon various intrinsic and extrinsic factors, and also that may be variable according to the stage of growth of an organism. Although genomically the same organism remains exactly same throughout its life cycle, but the proteome gets changed. So therefore, the overall variability in the living system may well be explained not merely by the variation in the genomic sequence or the gene sequence, but along with that, we shall have to also consider the variation in the proteome, the pool of proteins present. So the same organism at different stages may have the same genome but different proteomes. Therefore, the variability or the diversities in living system can only be explained if we go into depth of not only the genome but also of the proteome. But of course, there lies another aspect of the variation, as we have explained earlier, that although there may be presence of several genes in an organism, all genes are not expressed at the same time at the same rate. There may be variability in level of expression, and sometimes some genes can be expressed and sometimes some other genes can be expressed, or some genes can be expressed at the same rate at all different times. So there are great variability in the expression system also. And as we know, the expression of a gene starts with transcription. Transcription is the first stage in expression of a gene. So the product of transcription is a transcript or a messenger RNA. So the messenger RNA corresponding to a particular gene sequence contains the same genetic code. That means it is simply a complementary sequence of the DNA sequence of the gene, except for the fact that the variation is there in terms of the nucleotide sequence. That is, as we know, thymine is replaced by uracil, and second is the pentose sugar deoxyribose is replaced by ribose in messenger RNA. But it carries the same sequence of information or codons as was present in the original gene sequence or the DNA sequence. So therefore, if we study the messenger RNA sequence, we can immediately correlate it with the DNA sequence of the gene from which it has been generated, because it is complementary to each other. Secondly, if we find a messenger RNA corresponding to a particular gene sequence, it gives us an idea that that particular gene is in the process of expression, because transcription is going on. And the number of messenger RNA corresponding to a particular gene sequence also gives us the idea about the level of gene expression, because more is the frequency of transcription or rate of transcription, more will be the number of messenger RNA produced per unit time. So therefore, if we can study the messenger RNA and its number and sequence, it can very easily give us the idea about the process of gene expression. Therefore, there is another stage or level of study of the genetic mechanism of control of phenotypic characteristics in an individual, that is by studying the messenger RNA. And that particular aspect is known as transcriptomics, because like proteome, transcriptome refers to the pool of transcripts, pool of messenger RNA present in a particular organism at a given point of time under the influence of a given condition. So transcriptome also are variable in different times, in different stages of the life cycle, or maybe under the influence of different intrinsic and extrinsic conditions. So therefore, another level of study biology is the study of transcriptome, what we refer to as transcriptomics. Similarly, the overall study of the phenotypic characteristics or phenotypic behavior of an organism, what was conventionally discussed under the conventional biology, under zoology and botany, refer to the study of the variation based on the phenotypic characteristics. So that aspect might have only covered merely around, say, 20 to 25% of what is relevant to the variations or the overall diversities in the biological system. So traditional biology, which was based on the variability in the phenotypic characteristics, can be referred to as phenomics. So in omics technology, therefore, we have got several levels of understanding of biology. But of course, I shall have to also refer to another level of omics technology that we call as metabolomics. So what is metabolomics? As we have said, the variability in proteome can give us the idea of variability in phenotype. But as you know, proteins do not act standing alone. So proteins can interact among themselves, or proteins can interact with some other biomolecules, small molecules, or which are commonly referred to as ligands. And after a network of interactions among several proteins and other biomolecules, a particular biological activity or biological phenomenon can happen. And therefore, the mechanism of interaction between proteins and other biomolecules in a network-like fashion, which can be represented by metabolic interactions between biomolecules, can be referred to as metabolomics. And therefore, now we have in biology different levels of understanding of the biological diversity, starting from genomics, which is the fundamental basis, and then transcriptomics, which refers to the study of the messenger RNA, second level of understanding beyond genomics. Then proteomics, study at the level of all proteins. Then metabolomics, and then finally phenomics. So the traditional biology may have represented only a part of the understanding of biology today at the age of omics, which is actually the phenomics part that was traditionally covered by the conventional biology. But other parts have emerged only after the advent of genomics and other omics technologies. So therefore, our understanding of biological system will depend upon expanding our knowledge in all these levels of omics technology: genomics, transcriptomics, proteomics, metabolomics, and phenomics. And in this process, bioinformatics plays a very, very important role, as many of the intricate methods used in understanding the different levels of omics technology are highly complex. Therefore, it requires the use of computational method and information technology. Therefore, today, people have used our knowledge and understanding of principles and procedures of even the material system or material science to understand, analyze, and interpret the interaction between biomolecules. And there lies the importance of bioinformatics. So bioinformatics will give us the tools or the mechanisms or the methods based on which we can analyze and interpret the complex biological processes by use of not only the methods that are known in biology, but also by utilizing the understanding of material sciences and their applications. So therefore, bioinformatics is now a key to our understanding in modern biology. Without bioinformatics, we cannot think now of studying advanced biology in any stream. So there are many areas of bioinformatics which are emerging or evolving during the last 3 or 4 decades. As we said, that the journey of bioinformatics started only after the advent of omics technologies, particularly after the advent of genomics in the early part of 1970. And thereafter, there are many areas in which the bioinformatics are being applied. And newer areas are also emerging in the field of bioinformatics, which are being applied in understanding the different aspect of biological system. So major areas under bioinformatics are genomics, proteomics, drug designing, computational evolutionary biology, measuring biodiversity, and modeling biological systems. So genomics can be divided into two aspects: structural genomics and functional genomics. As we discussed earlier, the variability in living system can be partly explained by variation in the structure of the genome or the variation in the nucleotide sequence of the genes and genome. So that part is covered under structural genomics. So it is related to sequence analysis, genome annotation, and comparative genomics. So I think I need not explain sequence analysis. Already you know about it. What is genome annotation? Annotation is, say, in simple terms, it can be compared to labeling, you can say. That means annotation means establishing the role of different components of a gene or genome sequence in terms of its functional characteristics. That means finding out the gene sequences within a genome sequence. What are the coding sequences that we have to find out? Secondly, we have to find out which gene is responsible for production of which protein, and then correlate it with the biological activity or function. This is called as genome annotation. Then comparative genomics means comparing the genome sequences between organisms, maybe between strains of the same species or different species. So this is called comparative genomics. So overall, sequence analysis, genome annotation, and comparative genomics are important components of structural genomics. Similarly, the functional aspect of gene structure and the nucleotide sequence is covered by functional genomics. There are again many aspects of functional genomics like analysis of gene expression, analysis of regulation of gene expression, how the expression of a gene is regulated by genetic as well as epigenetic mechanisms. And the third is analysis of mutations in cancer or in relation to other pathologies. What is the role of mutation, or what is the specificity of a particular mutation with development of a particular type of pathological condition in different biological system. So mostly it refers to the mutations in cancer, but I think you might be also familiar to another term called polymorphism. So polymorphism and mutation differ in the sense that all mutations may not be occurring at a very high frequency. Those mutations which occur in high frequency, at least observed in 1% of a given population, they are usually referred to as polymorphism. We'll discuss about all these things in greater detail later on. The other aspect of bioinformatics application is proteomics, as I said. Although here also there are two types of analysis, one is structural and the other is functional, but we do not conventionally divide them into structural proteomics or functional proteomics, but rather it refers to the application of the knowledge of proteomics in analysis of protein expression and prediction of protein structure. We shall discuss about all these things a little bit in future classes. Then the drug designing. It is another field of activity in which bioinformatics plays a very, very important role. So earlier, to design and produce a new drug which is therapeutically useful and commercializable, it took around, say, 22 to 25 years by the conventional process. But after the advent of bioinformatics, many steps in the drug designing and drug discovery process can be achieved quickly, rapidly by use of computational method. So many computational methods have emerged which may replace some of the preliminary aspect of conventional drug designing process or drug discovery process. So therefore, the total time and cost involved in drug designing has come down to a greater scale. And now, within a period of 7 to 8 years, a new drug can come to the market because of the advent of various tools and techniques in bioinformatics, which has reduced the time required for the relevant processes which are involved in drug discovery method. We will discuss about drug designing and bioinformatics in a separate class in a little greater detail. Next is computational evolutionary biology. Evolution for millions of years have resulted in the development of different biological forms or biological systems, as we know. Therefore, it is essential to understand the evolutionary process by establishing linkages between different forms of living system based on their genotype or based on their genetic variation. So earlier, to the advent of the omics technology, people used to have the study of the genetic variation between species based on the phenotypic characteristics. But after the advent of the omics technology, now emphasis is given to establish relationship between species and to find out the variation between species based on the genomic as well as proteomic analysis. And that has resulted in better understanding of the evolutionary biology and to establish the phylogenetic relationship among different species. So this is called computational evolutionary biology. By application of bioinformatics, by computational method, we can now establish beyond doubt the relationship between species, which species has evolved from which species, and which species in evolutionary history is more closer to another species, and which are away in the evolutionary history from one species. So all these can be established by examining the variation in genotype or in the proteomics characteristics. So that is also we shall be discussing separately in a chapter involving phylogenetics or molecular phylogenetics, phylogeny, and phylogenetic analysis that we shall discuss separately, and also we shall do some practical so as to understand how bioinformatics tools can help us in drawing conclusion regarding evolutionary relationship between species. And that has got a very important role now, not only to find out variation among species, but also variation within species in terms of different serotypes, different biotypes, or different strains of the same organism. And this is one of the important mechanisms of another aspect of computational biology to establish the molecular epidemiology or to study the molecular epidemiology of different pathogens among viruses or bacteria. We can establish the variations and similarities between strains of the same organism relevant to different epidemiological situation. So that is called molecular epidemiology of pathogens. That also we shall discuss a little bit later on. So by that way, as I said, that use of bioinformatics can establish the evolutionary relationship among species. Therefore, we can also study the distance between species or the diversities within a species or among species can be measured or quantified by use of these bioinformatics tools. And biodiversity measurement, therefore, is a very important aspect of application of bioinformatics today. Now, application of bioinformatics has not limited it to only the areas that we have explained so far, but it has gone far beyond that. And our understanding of the genomic setup of an organism and understanding its functional aspect relating to the phenotypic variation has made it possible even to design the genome of an organism categorically as per the desire of the worker. So computational models can be generated by which we can also design a desired genomic structure. And it has gone beyond that now to understand how we can model the desired type of organism by application of the knowledge of an emerging area under bioinformatics that is called synthetic genomics. So we shall discuss about little bit later on about synthetic genomics also. Craig J. Venter, a well-known scientist, has established the method of modeling genome computationally, and by using chemical synthetic method, genome has been constituted or synthesized, and such synthetic genome in bacteria has been successfully incorporated into living microorganisms so as to ensure sustenance of the living microorganism. So in bacteria, it has been already done successfully, wherein a synthetic genome of the organism is introduced into enucleated bacteria, and after that, the bacterial species can be converted from one species to the other, and even after incorporation of the synthetic genome, the bacteria can also survive and carrying on all the life activities as that of a normal organism. So this area has emerged only after 2010, and which is called as synthetic genomics. So we shall little bit discuss about all these aspects in future classes. Now, the journey of bioinformatics started with the development of biological databases. As we discussed earlier, when the genome sequencing methodologies were introduced, after the sequences of various genomes were made available, it was quite challenging for the workers to to document, store, analyze, and interpret the vast or enormous quantity of data that have been generated by sequencing experiments. As we have already discussed, that one single genomic sequence of one single human being is of the length of 3,300 megabases. So if you start writing the whole genome sequence of one individual by keeping it in a note sheet or a book, I think several days will be required to note it down completely, because it's so lengthy a sequence. So not only documentation, but also in storage, analysis, and interpretation of such data, the manual methods were not found to be quite suitable. And therefore, people started using computers for storing those data and sharing among scientists. And thereafter, they tried to develop certain computational methods or tools so as to analyze the complex data derived out of these experiments. And that has initiated the process of emergence of the field of bioinformatics, as we discussed earlier. So biological databases are nothing but the storehouses or the libraries of life science information, which store the collected data which have been generated from various experiments conducted by use of genomics, proteomics, metabolomics, and other bioinformatics methodology. So they contain different kinds of information. Say, for example, some databases may contain information pertaining to the DNA sequences, some may have RNA sequences, some may have annotated sequences, some may have unannotated raw sequences, some may have sequences of the metabolic pathways. All these different kinds of databases are present. We shall take on in the next class the biological databases and their classification. Then we will discuss about what are the different types of biological databases, what is the role or importance of biological databases, and how they can help us in studying biology. So as I said, depending upon the type of information a biological database stores, they may be classified into different types, say primary sequence database, sequence database, protein classification database, pathway database, microarray database, two-dimensional polyacrylamide gel electrophoresis database or 2D page database, public literature search database, patent search database, etc. We shall discuss about all of them in the next class in greater detail. Out of all biological databases, there are thousands of databases now available over the net. Out of all the different databases available for the biological workers, there are three databases which have been used extensively worldwide, and they are the primary resources for our biologists. But interestingly, all these three resources were created by institutions located in three different continents of the world. They are GenBank, which is a research of all the resources available for a biologist, that is hosted by, NCBI, National Center for Biotechnology Information, which is managed by the National Institute of Health, government of US. Then another important database is EMBL, European Molecular Biology Database, or now it is referred to as ENA, European Nucleotide Archive, which is housed at European Bioinformatics Institute located in Great Britain. And a third of its kind is hosted in an Asian country, in Japan, which is known as DDBJ, DNA Data Bank of Japan. So these are the three important databases or resources for the biologist. And the importance of these databases worldwide have been recognized, and therefore consistently these three databases have been enriched by incorporation of data obtained from the experiments conducted globally at different countries and by uploading them to the databases. Most of the sequence information or the biological information that are generated from the experiments in European Union or European countries are mostly uploaded to EMBL or ENA, and some of the Asian countries that are uploaded to DDBJ, but most of the global community upload information to the database which is housed at NCBI, that is GenBank. It is pronounced as GenBank or GenBank. Many people pronounce it as GenBank. Now, these three databases are not standing alone now. So, realizing the importance of having the linkage between these three databases, they have been interlinked, and therefore now a particular biological worker can obtain the information from across these three databases while entering to one of them. So if we enter to GenBank, we can also have access to the information that is available in DDBJ or EMBL. So this is applicable to each of these three databases. When you enter to EMBL, from there, you can also have access to the information in GenBank and DDBJ. So they are integrated among each other. Now, let us discuss about some of the examples how bioinformatics has helped the biologist to solve their problem. I'll cite only one or two or three examples so as to understand the importance of bioinformatics in solving the problems in biology. Say, for example, one of the important problems to the biologist earlier while studying conventional biology is to find out the genes in a genomic sequence. When a genomic sequence is available for a particular organism, if you see the entire genomic sequence, each of the parts of the genomic sequences are not relevant to the coding part, or each part of the genome sequence is not involved in production of proteins and therefore not related to the phenotypic characteristics. In fact, most part of the genomic sequences of particularly eukaryotes are covered by non-genic or non-coding components rather than coding components. Coding sequences are occupying only a minor percentage of the entire genome sequence. Say, for example, in human being, only 2 to 3% of the entire genome sequence constitute the coding sequence. They are relevant to the phenotypic characteristics. But 97 to 98% of the entire genome sequence are not related functionally to any characteristics of the human being. They are non-coding sequences. At the beginning of understanding of genomics, people thought that these non-coding sequences existing in the genome sequence are irrelevant and they were referred to as junk sequences. But it was found later on that they are not really junk because the experiments conducted targeting deletion of certain parts of the coding sequences from the genome has resulted in non-functionality of the particular system, even death of the particular organism in some cases. So deletion of the non-coding part may result in, so to say, some interventions in the expression of genes, or may even result in complete stoppage of expression of certain genes, or even death of the particular organism. So therefore, they are not really junk. The non-coding sequences also play a vital role, not only in maintaining the structural integrity of the genome sequence, but also there are certain components of the non-coding sequence which are very important as regulatory components which control very precisely the expression of the genes. So therefore, it is very interesting and important to find out the coding sequences of a genome sequence, but there lies the complexity. As I said, out of a 3,300 megabase human genome, only 2 to 3% are coding sequences. How to locate or identify the coding sequences and how to differentiate them from the non-coding sequences? How to identify the genes? So gene finding was a big issue. Gene finding was a big problem at the beginning of the genomics era. So given a DNA sequence, how to predict the location of genes and how to locate within a gene sequence the coding part, that is exon, and the non-coding part, that is introns, in case of eukaryotes. It was a big issue. So this is done by, first of all, understanding the point that to be a gene sequence, a particular DNA sequence or the messenger RNA sequence obtained as a complementary sequence of the gene sequence must have to have a contiguous length of sequences containing functional codons without any stop codon in between. Why it is important? Let me explain a little bit. Normally, in a functional protein, in most of the organism, it has been found that most of them contain 200 or more amino acid residues. A functional protein mostly contain a number of amino acid residues. Of course, there are exceptions. There are few proteins which may have got only few amino acid residues. But in most cases, in most of the cases, you will find that a functional protein contains 200 or more amino acid residues contiguously arranged. So to give rise to amino acid residues in the protein by translation, the transcript or the messenger RNA must have to have a contiguous sequential arrangement of codons which code for different amino acids. And necessarily, there should not be any stop codons in between so that prematurely the translation stops. Let me explain it with the help of diagrams. As we know, there are 61 functional codons which are related to different amino acids, and there are 3 codons, triplet codons, which are known as stop codons. Stop codons do not code for any amino acid, and therefore it will result in stoppage of the synthesis of protein or the translation process. Now, suppose there is a small part of DNA sequence we want to understand whether this particular part of the DNA sequence is coding sequence or not. Okay. So, but of course, here for the sake of easy understanding and explanation, we have kept only 4 or 5 codons. But in fact, we shall have to study a long sequence having several hundreds of codons to find out a gene. I'm just giving an example by using a small piece of sequence. Say, for example, this is a part of the sequence of a messenger RNA. This can be read in one particular strand of the DNA sequence or one strand, for that matter, the messenger RNA in 3 different ways. You see, we do not know whether the codon starts from the first nucleotide of the sequence. Suppose if we consider that the first nucleotide in the part of the sequence is a part only. This is not the complete gene. It's a part only. We do not know whether this particular nucleotide, first nucleotide, is a part of the first codon as the first nucleotide, or it may be a part of the previous codon, or it may be the second nucleotide in a codon or third nucleotide in a codon. That we do not know. Therefore, there are 3 possibilities of reading this particular sequence. This is one where we consider the first nucleotide as the part of the first codon, the first nucleotide of the first codon, and therefore the first codon will be CUC, second codon will be AGC, third will be GUU, and the fourth will be ACC. So in that case, if this is true for this particular part, then it may give rise to this particular peptide sequence in the protein after translation. CUC stands for leucine, AGC stands for serine, GU stands for valine, and ACC stands for threonine. So the adjacent amino acid sequences of the peptide sequence will be leucine, serine, valine, and threonine. But if we consider that the first nucleotide is a part of the previous codon, it's not a part of the first codon, then we can start reading the sequence from the second nucleotide. Then the first codon will be UCA, second will be GCG, like that. It will go on. All codons will differ from the previous one. So in this frame, if we study or if we consider the sequence, the second frame will give rise to a different peptide sequence: serine, alanine, leucine, and proline. Similarly, if we consider that the first 2 nucleotides are the part of the previous codon, only the third nucleotide is the starting codon, starting nucleotide of the next codon, then the codons present in this particular sequence will be for glutamine, arginine, tyrosine, and histidine. So therefore, you see, depending upon the reading frame, how do you read the sequence, the interpretation of our amino acid sequence will be different. If the reading frame is this one, then amino sequence will be this one. If the reading frame is changed, the amino sequence gets changed. So there can be 3 reading frames in a particular part of the DNA sequence, or for that matter, messenger RNA. But out of these 3 reading frames, only one is functional in an organism. Okay? And one only constitute the coding sequence for the particular gene. If it is a gene sequence, only one of these reading frames will be really coding for a particular protein, if it is a gene sequence. So for that matter, we shall have to find out what is the property of such reading frames. Out of these 3 frames, we can designate one of them as open reading frames. What is an open reading frame? ORF. ORF is a reading frame consisting of triplet codons sequentially arranged in a messenger RNA for consistently a long length of the sequence without having any stop codon in between. Suppose in this particular frame, after 3 or 4 codons for amino acid, there appears one stop codon. What does it mean? Means while it is being translated, after the messenger, the ribosome reaches the stop codon in the messenger RNA, there will be stoppage of adding more amino acid to the peptide chain. But an amino acid sequence consisting of only 4 or 5 nucleotides, amino acids will not constitute a functional protein. As we know, a functional protein contains a sufficiently long peptide chain consisting of about 200 or more amino acid residues. So therefore, if it prematurely stops synthesizing a peptide chain of sufficient length, it cannot represent a particular functional protein. So it can be regarded as a functional protein if it contains 200 or more amino acid residues in it. Therefore, in the gene sequence, there should be contiguously present at least 200 or more functional codons without stop codon in between. And that type of sequences can be regarded as real coding sequences, and they are referred to as the open reading frames. So what we do to find out a coding sequence, we first read the sequence in 3 different frames and find out which particular frame contains no stop codon in between for sufficient length so that it can represent a coding sequence. So that is first and foremost property of a gene. Then we shall have to find out the initial codon, that is your AUG in the messenger RNA, because in all amino acid sequences or protein sequences, methionine constitute the first amino acid residues. So AU is the start codon, and then we can identify that this is the start codon, and towards the upstream of that start codon, there will be the transcription start site and the promoter sequences. And they can also be identified by identifying the specific sequences involved with the promoter sequences and the transcription start site. And therefore, we can finally identify the gene sequences by doing this. So first and foremost idea of finding coding sequences or gene sequences is to find out open reading frames in a particular length of genomic sequence. So I think now you can understand how genes can be identified by identifying open reading frames. So by employing these methods, various software tools have been developed, and they can be used now to determine the gene sequences of a genome. So some examples are GeneMark, Fzine B, Glymer, or you can also use one online server, www.genefinding.org. You can put up your genomic sequence into it, and it will give you the ORFs and identify the genes in the sequence and differentiate the non-coding part from the coding part of the sequence. So here we stop for today. I think everything is clear to you so far, whatever we have discussed today.